So what we've developed is a 91 gene classifier that predicts which women diagnosed with hormone receptor positive early stage breast cancer would recur earlier, meaning within 3 years of diagnosis, versus later, meaning 10 years, or beyond from the time of diagnosis. And these recurrences are distant metastases. So although we can predict that, you're right, we actually don't know what we can do about it. So the question is, now that we've identified some of the genes that make up that classifier, can we take advantage of that and develop clinical trials targeting the gene products in addition to tamoxifen or maybe the aromatase inhibitors to do better than we're already doing in treating these women? So it would depend on how we develop the clinical trial. You know, typically these combinations of endocrine therapy and biologics are done in metastatic breast cancer, which is completely appropriate. But now that we've identified a means of collecting subsets of patients that might benefit from endocrine therapy plus biologic, we can hopefully develop these trials in earlier stage disease and do better in terms of curing these women. Right, so we have multiple gene classifiers for different subsets of women with breast cancer, and this again is specifically for early versus late recurrences in hormone receptor positive disease. I think the interesting part about what we've identified is that many of these genes in the classifier actually make a lot of sense, right? So epidermal growth factor receptor, BCL2, SRC, the estrogen receptor, androgen receptor, these are known genes that are associated with breast cancer in some shape or form, and importantly already have drugs that target their gene products. So we have the ability pretty soon, in fact, to do clinical trials incorporating those agents intelligently with endocrine therapy.